So the next talk is going to get a little bit technical. And if you're just happening in the area of particle size characterization, and you're lucky because you will get a lot of information. Now, hopefully, after the presentation, you will look at the slides because I do put a lot of information over there. For the time constraint, I may not be able to cover all of them, but the examples, the references are all in the slides. And for those of you who are not necessarily in this area, sit back and relax, but don't sleep. And for those who are online, don't walk away. And hopefully you find this talk interesting. So the title is pretty long, but what we'll focus on is particle size, and in the context of product quality and bioequivalence. And talk about product quality. I think you are pretty familiar by now. What is a pharmaceutical quality? A quality product of any kind consistently meets the expectation of the user, and drugs are no different. And patients expect safe and effective medicine with every single dose that they take. And pharmaceutical quality is what's assuring that every dose is safe, effective, free of contamination and defects, and it is what's giving patient the confidence in their next dose, not previous dose, but in their next dose that they're going to take. Talk about the Office of Pharmaceutical Quality. I'm pretty sure you're aware that OPQ is part of the reviews or assessing your application. So this is probably you often interact the most. But you probably are not aware of is that OPQ has a very robust science and research program. So we do have our own laboratories on campus, and then to help solving some of the challenging regulatory problems. So the science program is really designed to be prepared, be ready when there is emerging health issue. For example, heparin, valsartan, where nitrosamine are impurities involved, or opioid product that with abuse deterrent formulation. So those are the new areas of science that we need to be ready. But how can we be ready if suddenly it appears? So then we need to have a really robust research program to anticipate what will be the problem down the road. So it has to be forward-looking. So in terms of research program we have in-house, we're focusing on new technologies to look at how to improve the analytics and manufacturing. For example, 3D printing, continuous manufacturing. Those are examples of the emerging technologies that can be used to help improve the quality of the product. And of course, advanced analytics. So I will talk some example in this presentation. And also, when the new drugs is approved, we don't wait until the time of generic application comes in, because if there's a gap being identified, there is a need in scientific research need to be filling those gaps, then we do conduct research in-house and to help understand those, and later on can help with the review divisions to conduct the reviews. And the complex generics, of course, that is what this workshop is about. In terms of the lab, as I mentioned, we have lab in the FDA, and we interact with the review offices through lab consults. So helping with massive verification, evaluating product quality, and more relevant for this talk, that we do work with OGD quite frequently to evaluate whether method can be used for determination or in support of the bioequivalence. And the knowledge that we have gained through our own research, we do not just hold with ourselves, we also provide trainings to the reviewers. So just last week, just coincidentally, last week we finished 2-day training to reviewers on the topic of particle characterization. So that is 2-day training. Today we're going to talk about for 20 minutes. So it's a really condensed material. And also the research will eventually will translate to some of the guidances, because that's how it should be used, and to elevate the science. And then in-house we generate data to support that. So with that background mentioned, so we will move into the real topic of today. So I'll explain to you the why. Why is particle size important, and why it is challenging, and how do we do it, and what are the common deficiencies we've seen and I'll explain them using examples. Now the why. If you are developing product and if you're seeing particle size as part of the specification, and you know it's an important pharmaceutical quality, critical quality attribute, certainly you need to have good method to measure that. So for any dispersion product, emulsion, suspension, liposomes, iron colloids, and also this morning talk about some of the transdermal topical product, whenever there is a particle presence, you need to have a good particle size measurement as part of the quality to ensure the quality. But in recent years, because of difficulties with bioequivalence, some of the complex generics, we've seen the trend is utilizing some of the in vitro characterization tools to help in support of the bioequivalence determination. And here are the two examples of the PSG guidance mentioned about particle size characterization or globule size characterization. We use them interchangeably, globule and particle. And as you can see, as you can imagine, there are a variety of particles that's present in pharmaceutical product, and they can vary in size, as small as a few nanometer and as big as a few hundred micron or a few millimeter if you're talking about the powder material. So really it's quite diverse. So there comes the challenge. How do we characterize the size of such a diverse type of material? And also, what is typically misunderstood about the particle size characterization is what we are generally want to know about the particle size. What we are really interested is actually not the particle size. It's not the size of the particles, but what we are really interested is what the particle does. So it's not an objective to measure the particle size, but it's a means to an end. And that means, for example, physical stability, if the particle to settle down, if the particle really making nanoparticles, then increasing surface area to enhance the dissolution. So we care about particle size is because we care about how it's being utilized in the drug product. And also in this context, bioequivalence. So the objective of the particle, or what we understand the purpose of measuring particle size, dictate how should we measure it. That's one thing probably misunderstood. But once we focusing on the measurement of the particle size, there's another challenge that many of the conditions, quite surprisingly, that they can influence the measurement results. The sampling preparation procedure, the instrument choice, and all of them can impact the particle size measurement results. So if that happens, as a developer, you certainly want to generating more reliable data, and from the agency's perspective, we want to make sure you understand what are the risks, and then you're taking steps to mitigate those risks, and you have a proper control of measuring the particle size. And the how do we measure particle size? And this is a table of not complete, a list of instruments or techniques that you can use to characterize the particle size. And changing from very small to really large, and for the liquid dispersion to the powder material. And they vary quite diverse in terms of the size range that they can measure. And my colleagues in OPQ IO science staff, recently they have done a survey of the nanomaterial as approved. They look at what are the commonly used techniques. Well, they found out that for the nanomaterial, dynamic light scattering is most commonly used technique. About 50%. And then there's laser diffraction, which is another type of light scattering measurement. And then there's microscopy. So this doesn't mean that light scattering, dynamic light scattering is the best method to utilize, but for certain range of the product, there's a certain technique that's better suited to measure the size. So what are the points to consider when you are developing a method for measuring the size? One is about the sensitivity of the method. For example, if you are changing the manufacturing process, and you know the manufacturing process, a milling procedure or a homogenization procedure, the procedure can impact the particle size, and you certainly want to have a particle size measurement method that is able to detect the differences because of the manufacturing process changes. So sensitivity, you need to demonstrate that is able to detect the differences, intentional changes in the process. And the specificity, we don't mean here in the same way as ICHQ2R1 in terms of validation, because as you may know, particle size measurement is non-specific. So you do not know what you are measuring. You only know what the outcome, but you don't necessarily know this particle is from this and this size is from that. So what we mean the specificity as part of you are developing the method is so that in case there is a presence of the interference, that you are not measuring the interference. Because if the API is what you are interested, and if the polymer that's interfering, you certainly don't want to measure the polymer. You want to measure the API. So how do you take care of that? That's part of the method development and the validation. And also you have the choice to make whether you choose one technique or multiple techniques. This is commonly dependent on what instrument you have. But the more instrument you have, you can understand better in terms of the limitations of each instrument. So that can give you a better idea in terms of totality of the information about that particular product. So some of the common deficiencies, I don't think I will have time to go over each one of them in terms of why they are deficient. But first, of course, if you're using wrong instrument to measure the wrong particles, colloid iron is really small, less than 30 nanometer, so you cannot use a laser diffraction. Micron size range more suited for that measurement. And particularly for the dynamic light scattering, if you are reporting the results, if you are reporting in distribution analysis, you're using distribution analysis to report, it involves the conversion of the intensity-weighted data to volume-weighted, to number-weighted, and also involving converting the raw data, which is intensity, to some other number, which you need to know everything about optical property of the material. Most of the time, it's very difficult to have all those information. So if you're using DLS to report and distribution results, it's better to put correct dispersive refractive index, and also what is the algorithm analysis used to conduct or report those values. Intensity instrument for the dynamic light scattering, it generates intensity-weighted results. So it's not appropriate to convert them to volume-weighted, to number-weighted, because that carries high uncertainty and the risk of error propagation. So stay with intensity would be safer. And what to validate and what material you use for validation. This is very common. We have seen as deficient is the company use the size standards, known size standards, to validate in terms of the accuracy. However, we know the polystyrene standards, for example, is very different from a drug product. Emulsion, for example, the properties are different, the methods for dispersion are different, and the parameter for instrument to measure are different. So even if you validate the particle size measurement using a known size standard, a NIST standard, for example, that still does not give you or give us confidence that the method is necessarily appropriate for the sample that you're going to measure, which is your sample. So to do the validation, it's better to use the actual sample to conduct the validation. And you can use size standards to qualify the instrument, but not for the purpose of validation. What it needs to be validated is precision, the robustness of the method. I'll talk about that later. And also some of the method detail is really important to be included because this will allow us to assess them more complete. So next few slides, I'll give an example of what can go wrong. And I'll focus first on the dispersion medium. This is sometimes misunderstood because when we talk about particles, we generally pay attention to what is dispersed, a particle and globule. And for example, emulsion here, an oil globule with a surfactant, we generally pay attention to what is dispersed. And another example of suspension. What we didn't necessarily pay attention to is the environment that they are in. But those environment is quite important for the stability of those dispersed system. For an emulsion, if you dilute, for particles, suspension, if you dilute, what's going to happen? So when you change the native environment of the particle, it may undergo, for example, putting a powder into a liquid, it may dissolve and it may aggregate. And those characteristics will change the approach that you should be taken to develop a size measurement procedure and method, and also changes how you should be analyzing or interpreting the results. So now I'll give you a real example. This is a real example generated in our lab. This is ophthalmic suspension drug product. What we found out, this is using laser diffraction, by the way. What we found out that once we introduce material to the instrument, over time there's a decrease in the light obscuration. So this is basically the signal or the concentration seems to be decreasing over time. At the same time, we observe the appearance of secondary small peak that is less than 1 micron, and the bigger peak is what's original. So over time, we saw a decrease in the obscuration signal, as well as the appearance of a smaller population of the particles. And the third clue is that we saw the average, the volume-weighted mean particle size is decreasing over time, or decreasing with further dilutions. So what is happening? Is that two bimodal distribution real? What we, of course, did some of the evaluation. What we found out that the particles, of course, can dissolve, as I mentioned. And we knew that beforehand. So we saturated the API, or we saturate solution with the API, hoping that that can solve the saturation problem. So the product should, or API should not be dissolving. However, what we noticed that is this particular API tends to cluster and does not disperse well in the medium. So what's happening is particle is not wetted properly. So the wetting is not done well, so the saturation is actually not achieved. So then what we did is add a little bit of surfactant, tiny amount, just above the critical micelle concentration, and sonicated of that API, and then we filtered out and get the solution, and then ran the laser diffraction, which we get a single distribution of the larger population. So this is an example that what you may encounter as you are developing a method, once you know what can happen, and you pay attention and utilize that information to help you develop a good method. So the next about the evaluation. In terms of validation, what do you validate? Do you need to validate the accuracy, the trueness? This is from the USP 776. This is a particle in black circle. And you can see there are many different ways to describe that particle. And this is a two-dimensional object. If we talk about a real-life particle, it's a three-dimensional object, meaning you have many different ways to describe what is the dimension of the particle. So what is a true size? It depends really on what you are describing it. And surprisingly, for particle size characterization, when we're measuring particle size, we're never measuring actually the size. We are measuring some property that is related to size. And this is a concept of equivalent spheres. And for example, if we are measuring this particle irregular shape, we measure how fast it diffuses in a liquid medium, and using dynamic light scattering, then we're measuring hydrodynamic radius. We're measuring how fast it's diffusing. If we're measuring by sieving analysis, we're measuring how does a particle go through a sieve, a pore, and then going to the below the sieve. So we're measuring the minimum dimension of that particle, not the length of the particle. We're measuring the minimum length. So depending on the technique that you're using, what number you are reporting actually changes. So it's really difficult to define what is true. And also to go back to the purpose of your measurement. If your purpose is to make sure the process is capable, or the dissolution is the two particle size changes dissolution behavior, because of that particular purpose or objective, you need to evaluate the size in terms of tolerance of the accuracy differently based on the purpose of the use of the size values. But what is important for the validation is the precision. So it's really important to make sure that you can demonstrate that method is precise and robust for its intended purpose. And what are the typical components of the validation? What needs to validate? Precision, repeatability. So this is exact same material in the instrument and then measure multiple times. Nothing changes, only the time. And this typically gives you information whether machine is working properly, whether sample has stability problem. And remember the example I just gave a few slides ago, that about the decreasing in the obscuration, and that is a perfect example to show how do you use repeatability measurement to give you some information about what's happening with that particular particle. Reproducibility, this may involve using a different sample preparation. So you prepare multiple times and maybe by a different analyst, a different day, and or using a similar but not identical instrument. And this gives you information also about sampling procedure, dispersion, and also whether the machine is working properly. And the robustness. So you need to intentionally introduce some changes, and you need to be able to see the change. So that gives us confidence that this is a good method because it's supposed to give you that information. In terms of dilution, because this typically come up during the method development, for dynamic light scattering and laser diffraction, this is very common, but they appear at the different stages. For the dynamic light scattering, usually the dilution occurs before the measurement. So you prepare material, you dilute them. And for the laser diffraction measurement, the dilution occurs during the measurement. So those two dilution steps are different. But what can happen in a dilution, when you dilute the sample, usually there are two aspects. One is when you're diluting it, the particle actually change, dissolve, for example. Or in this case, we're measuring a polystyrene beads. We know it should be 100 nanometer, but we use water to measure it. It gives about 110 nanometer. But if we use sodium chloride, introduce some of the ions to suppress the hydration sphere, then we can get a more precise measurement when closer to its true number, it's 100 nanometer. And in the other example, when we dilute, what we may change is the environment of the particle. For example, this is a cyclosporine ophthalmic emulsion. We know when we dilute this emulsion, the polymer and the environment is different. So it changes how the particle move around. So this is by changing the environment, we can find the shift in the distribution of particle size as a result by simply diluting it. And the method interference. So this is what we found in recent examples that based on the experience, we found repeating a theme that because in the pharmaceutical drug product, we have the polymers, we have excipient, that we consider them inactive. However, they do have some role, or they can change how you measure the particle size. For example, in this example, we know our API should be around 5 micron, but in the presence of the polymer, we cannot accurately measure. Well, we see 2 peaks, and the 1 peak that highlighted in red, that is due to the polymer interference, polymer scattering. And with known size standards, we can show there is a presence of interference. And how do we take care of the interference? We prepared a placebo formulation, and using the placebo formulation to blank the scattering signal, and this gave us a clean signal of the distribution. I'm running out of the time, so I'm going to quick. This is the last slides about example. So what I mentioned about before, there are many different particle size measurements. But doesn't mean that all the methods available are perfect. So we need to look for better method. So in this example, we're using asymmetric flow, field flow fractionation to analyzing a relatively polydispersed material. We separate them first, and we analyze size by dynamic light scattering, and we're able to achieve a better understanding of what are the particles present, what are the globules present in this emulsion product, which provided better understanding for why all the numbers that we generated from different techniques are different. And in summary, I hope you agree with me that particle size is one of the critical quality attributes that are also important for the bioequivalence determination. And do not underestimate the challenges of characterizing the particle size. Yes, it sounds very simple, putting into the machine, generate a number, but it's far from simple. It's very complex. And also, every particle size instrument has its own limitation and the strengths, so we do need to pay attention. Therefore, we're looking for a better technique that can give better information. But regardless of the method that you're using, you have to make sure that the method is properly developed and properly or adequately validated in order to demonstrate it's suitable for its intended purpose. And in order for the reviewers to give more complete assessment, really needs the complete and full detailed information, sampling procedure, instrument parameters. So really important to include all those information in your submission, which will allow a good assessment. So with that, thank you for your attention, and I'll be answering your questions during the panel discussion.